Three types of digestive enzymes: amylase, protease, lipase. Yes, you can call it carbohydrase as well. Carbohydrase. An example of carbohydrase is amylase. You get marks for both. Protease and lipase. So you can tell by the name what they break down. Carbohydrase will break down carbohydrates. Protease will break down proteins. Lipase will break down lipids or fats. What do they break down into? So carbohydrates, if you were to break down carbohydrates, what would that break down into? Carbohydrates into sugars. Yep, it breaks down into simple sugars. Amazing. Or glucose, you can say glucose as well. Protease breaks down proteins into amino acids. Good. And we got lipids or fats. What do fats break down into? Exam boards, this is relevant to AQA, Edexcel, and OCR. Lipids breaks down into glycerols and fatty acids. So two things. Glycerol is just one of them. It's two things. Fatty acids and glycerol. Amazing. Let's get into the question. So I think some of you told me the answer for the first one. Where is amylase produced in the human body? Amylase. What do you guys think? What's the difference between glycerol and glycogen? Glycogen is a stored form of glucose. Glycerol is not. Glycerol is just what fats break down into. Salivary glands and pancreas. Yes. Not liver and pancreas. Liver produces something else. Liver produces bile. That's used to break down fats into smaller droplets. We call it emulsification. It breaks down fats into smaller droplets. It's not the same as lipase. Let's write that down here as well. It's not an enzyme. Bile produced by the liver, and it breaks down fats. We say it emulsifies fats into smaller droplets, and it has a secondary function as well. So that's not the only thing it does. It also, yes, amazing. It also neutralizes acids from the stomach. So you see that stomach, it has hydrochloric acid in it. So when the food comes out of the stomach, it's going to be really acidic. It's bad for the rest of the body. So we need to neutralize it to make it neutral again. So it neutralizes acids from the stomach. So that's what the bile does in the digestive system. Okay. Yeah. So it's not liver, it's not stomach. Stomach doesn't produce amylase, that produces hydrochloric acid. Salivary glands produce saliva, that's got amylase in it. And pancreas is another thing, it produces all three types of enzymes. Okay, next one. Enzymes speed up chemical reactions. Explain how amylase breaks down starch for 3 marks, meaning we need 1, 2, 3 points. What do you guys think? So it's basically asking, how does an enzyme work? Yes, lock and key. We need to talk about the lock and key mechanism. Yep. So lock and key mechanism. So we've got an enzyme. The enzyme is like, it's a protein. It's got like a gap in it. What's the name of the gap? I'm just going to draw it in this shape. What's the name of the gap inside? Active site. Yep, amazing. And we've also got, so this gap here is called the active site. And then what goes into the active site is the substrate. So substrate is the molecule that's being reacted. So let's say this is the substrate. It's a T-shaped substrate because that's a T-shaped active site. But loads of different substrates are present. So you might have this substrate here. You might have a circular one. These two, they're not going to fit into this active site. So this enzyme here has a specific shape. It's specific to this one here. So we got 1, 2, 3 different substrates. But this enzyme will only work with substrate number 2 because it's a complementary shape. It's the lock and key mechanism because this is like a padlock. This is like a key. These are keys for different locks. So these are not going to work with this enzyme. So what happens? This goes into the enzyme. What happens after it goes in? So let me draw another diagram of all of this inside. What happens once it's inside? Let me get a different color. It breaks it down. Yeah, the substrate. So the substrate gets broken down. So this fits into here and then it gets broken down into like maybe 2 pieces. So let me break it down into 2 pieces here. 1, 2. It's basically broken the bonds in between the substrate. So it's broken bonds here and it's broken it into 2 different molecules. Those are the products and they leave. They're done. So now it has space for another substrate to come in and this just keeps on happening over and over again. For GCSE, yeah, always 2 molecules. Yeah, you can, it always forms two molecules. That's fine. But it's more complicated than that, but you don't need to know all of that detail. So let's try and apply this to this question. 3 marks. Enzymes speed up chemical reactions. Explain how amylase breaks down starch. So what's the substrate here? What's the substrate in this question? Can you draw diagrams to explain answers? If they don't tell you to, I wouldn't. For this, I wouldn't. I think you should write words. For this, you shouldn't draw diagrams. So the substrate is the starch. That's the thing that's being broken down. Okay, so that's the substrate. And this is the enzyme. So inside the amylase, you have an active site and that's the gap here. And then you've got the substrate or the starch coming in. So you get 1 mark to say the substrate fits into the active site. Second mark, what do you guys think the second mark is for? Binds is fine. Yep, you can say that as well. What would be the second mark? Breaks it down. I'd say that's the third mark, to say it breaks it down. Second mark is to say that it's complementary. So this only works for starch. So the shape of the active site is unique to starch. So shape of active site is unique to starch, or you can say it's complementary. That's the second mark. And then you can say it's broken down. Starch is broken down to, like we said, sugars, simple sugars. Because this is a carbohydrate, carbohydrates breaks down carbohydrates into simple sugars. Are bullet points allowed? Yes, bullet points are allowed. I recommend bullet points in answering these because it makes it easier for the markers to mark them. This is relevant for triple and for combined as well. This particular question is AQA, but it's relevant to everyone. All right, let's move on. One sugar in the body is glucose. Glucose is used for respiration. Give one other use for glucose in the body. What do you guys say? Can you say products instead of sugar? Yes, you can. So energy wouldn't get you marks because it's already mentioned respiration. Respiration is used for energy release. So you wouldn't get marks for saying energy. What other uses are there? Yeah, you can store it. So you can't say contraction, you can't say movement. You got to be a bit more specific. What does the actual glucose turn into when it's stored? What do you guys think? What does it get stored as? Yeah, there we go. Someone said it. Wow, these comments are coming fast. Storage to glycogen. Glucose gets converted into glycogen. So that's don't get that confused with glycerol. So that comes under insulin. So this is under the hormones topic. So insulin, what that does is it converts glucose in the blood into glycogen. So these all sound the same, so don't get them confused. Glycogen is just a stored form of glucose. Glycerol is what lipase or fats are broken down into. So what you can say for this question, another use is stored as glycogen. Muscle contraction, again, that wouldn't get you marks because that's all to do with respiration. That's all to do with energy. So what they're looking for in this question is storing. Stored as glycogen, not stored as glucose. You could say used to make amino acids as well. Yep. And you can also say stored as fat. So it might be converted to fat and stored as fat. Just saying storing is not enough. No, you'd have to say what it's stored into. Stored as fat is fine. Yep. You can't say cellulose. Cellulose is just for plants. Animal cells don't have cellulose. Okay. Moving on. So this question's about diseases. It's not paper one for everyone. It's paper one for AQA. I'm not sure about, I think for EdXL might be paper 2 or OCR. I'm not entirely sure. Pathogens make us feel ill. Why do they make us feel ill? They release toxins. Yes. These are higher questions, but some of it might be relevant to foundation. So you can say release toxins. You can also say they damage cells. Yeah, that's fine as well. Or kill cells. That's okay as well. White blood cells produce antibodies. This is one way white blood cells protect us against pathogens. 2 other ways. There's 3 ways that white blood cells protect us. Do you guys remember what they are? If you watch my videos, you would know these. The A, one of them is for antitoxins. Yeah. We got phagocytosis. And antibodies, yeah. Antitoxins, these just neutralize toxins. So toxins is what's made, what the pathogens make. Phagocytosis, this is when you get white blood cells which engulf or eat or digest pathogens. We'll say engulf, that's the key word there that you got to use. And then we've got antibodies. So people saying memory cell, yeah, memory cell is a type of white blood cell. It's a lymphocyte which has basically survived after infection. So antibodies, what they do is they bind to the edge of a pathogen. We call those antigen. They bind to antigens on pathogens. And basically cause them to get eaten by phagocytes. So they basically mark the pathogens and tell the phagocytes to come and eat the pathogen, basically. So yeah, so we went through these questions last week. So all the videos that we did, all the lives that we did the last few weeks, we went through health and disease last week. They're all on YouTube, guys. I've uploaded them. So this one here, so if you go in Flash Revision Lab, we got one on health and fighting diseases. We got nervous system, where basically I'm going through loads of questions, and you guys can go onto this and you can actually print off the questions yourselves and you can try it with the video. So if you go onto any of these videos, then you can basically go to the comments and you can download the worksheet that I'm going through, and you can do it as you watch the video. So yeah, check that out if you want some more revision. Back to the question. So out of those three, we've got white blood cells produce antibodies. So they've talked about APA. They've talked about this one. We just need to mention these two. So 2 marks here. We've got they produce antitoxins. And the P is for phagocytosis. This is a mix. It's not just one paper, guys. It's a mix of loads of different questions that I've got from different papers. Vaccination can protect us from diseases pathogens cause. One type of virus causes measles. Doctor vaccinates a child against measles. What does the doctor inject into a child to make the child immune to measles? Inactive pathogens, yeah. So whenever someone gets vaccinated, what they're doing is they're injecting the pathogens into your body, but it's an inactive or a dead version of it. So it won't actually harm you, but your body thinks it's a real pathogen. So it starts to create an immune response to kill it. I I will post this on YouTube. Yeah, I will try and post this tonight so that tomorrow, because I know it's the last day before exams, so you guys can rewatch this and I'll upload all of these questions so you guys can try it again after. So hopefully by tomorrow it'll be up on YouTube. You can say inactive, you can say dead, you can say weakened. Yep, you can say all of them. Yep. I don't think this is an old question. I don't think they'll give you 2 marks for that. I think that's probably a 1 mark question, to be honest with you nowadays. It's quite an old question. Inactive pathogens. Altered, no, you wouldn't get a mark for altered. You have to say weakened, inactive, or dead. Yeah. Okay, next question. A few weeks after vaccination, the child becomes infected with measles from another person. The graph shows the number of measles antibodies in the child's blood before the vaccination and after the infection. Okay, so here the child got vaccinated. You can see the number of antibodies in the blood increases, then it goes back down, and then they catch measles, or they get the pathogen from someone, and then it goes up again. So the question, 3 marks. More measles antibodies are produced after the infection than after the vaccination. Describe other differences in antibody production after infection compared with after vaccinations. 3 points. There's one that loads of people say, but it doesn't actually get your mark. What do you guys think? You don't need to mention memory cells here. It is the memory cells which are causing this. You're right, but this is just asking to describe the differences. So when it doesn't say to explain. If it says to explain, that's when you got to use your theory. This is just asking you to describe the differences between this and this. This, again, guys, it's not a full paper. I've taken this from combined, I've taken this for triple, but this question is for combined and for triple. It'll be relevant to both. Okay, so I'm seeing a lot of people saying more antibodies. You don't get a mark for saying more antibodies. This is a common mistake because they've told you in the question, more antibodies are produced after infection. If you can't state anything in the question, they've literally told you that, so you don't get a mark for that. So there's 3 other things you need to say. Okay, so someone said steeper gradient. Good. If it's a steeper gradient, what does that mean here? See here, this line is much steeper than this line here. Good. Yeah, it means it's being released faster. So one difference, so we can say steeper gradient. Because it's produced faster. Quicker response. Excellent. Yeah, look at this. Look at where it's been vaccinated. Look at where it's been infected. See where it starts to rise. See here. It takes about 1, 2, 3, 4, 5 squares before it starts to rise. This one, it literally only takes 2 squares. So the rise starts much quicker after infection than after vaccination. So you can say quicker response. Or quicker response before the rise. And we got one more. Yes, lasts longer. Good. So this is when you got to look at the peak. So once it's peaked, what happens after the peak? See that? That's where the maximum antibodies are. See here, it goes back down really quickly. Compared to this starts to go down quite slowly. So this lasts longer. Doesn't drop as fast, so it lasts longer. That's another one. Any questions on that? I'm not sure if it's foundation or higher, guys. This is definitely higher. I'm not sure if it's relevant to foundation, but it's definitely relevant to combined and for triple. What lasts longer? The time that the antibodies are being produced at a high rate. So you see here, there's a lot of antibodies produced and it stays high for a very long time. Here, it drops back to normal. The amount of antibodies drops back to normal very quickly. Okay. Can I explain? Quick response? Yes. So look at where the vaccinations happened, look at where the infections happen, and look at how long it takes before the number of antibodies start to rise. See here, it takes 5 small squares before it starts to rise. Here, it only takes 2 squares. It only takes like this long for it to rise. Okay, hope that makes sense. Any other questions? I will go back to the info above. Don't worry, I'll give you guys a chance to screenshot all of this. Okay, so next question. Vaccinations against measles will not protect the child against rubella virus. So this is just another virus. Why not? For 1 mark. It's not paper 1 for everyone. No, but it is for AQA. Because antibodies are specific. Yep, good. So that means it's just like enzymes. See, enzymes, they're a specific. This one only binds with this one. Same with antibodies. One antibody will only fight against one type of pathogen. That's because of the antigen on their surface. Let me use a different color here. So here's a pathogen. Antigens are what's on the surface. It's like a marker that they have. And different pathogens have different shaped markers. So what an antibody does, this antibody would fit onto this marker, but if it's like this shape, this wouldn't because it's a square shape. So it's specific. So antibodies are specific to pathogens. So antigens, the difference, antibody is what our white blood cells produce to kill the pathogen or to attach to the pathogen. Antigens are these things here. They're markers on the pathogen. So whenever our body sees these pathogens, it decides to produce antibodies because it wants to fight it. So when you're getting vaccinated, remember, we inject dead pathogens into our bodies. Our body thinks this is a real pathogen, even though it's dead. Because it sees the antigens, it thinks it's a real pathogen. So it starts to produce these antibodies to try and kill it. So we get that immune response, and then what happens is those white blood cells that produce that antibody stay behind. So after you've cleared the infection, they stay behind, and that's what we call memory cells. So white blood cells which stay behind after infection, they're memory cells, so that if this pathogen was to ever enter the body again, it would be able to rapidly produce these antibodies to kill it. So yeah, if you want to know about this in more detail, check out the live from last week, the health and disease one. I think we go through this in that. We spend more time on that topic. So do that. What's the advantage of vaccinating large proportion of the population against measles? Yeah, herd immunity basically, yeah, reduces the spread of infection. Or of pathogen, which is the same as saying herd immunity. You wouldn't get a mark for just saying immune, no. This question's AQA. You can't just say no. You can't just say immunity. You can say herd immunity, but I would say reduce the spread of pathogen. That's what it says on most mark schemes. It doesn't usually say herd immunity. Herd immunity is just when a lot of the population are immune to it, so they're less likely to spread it. Yeah, less people will get the virus. Because less people have it, there's less people to spread it. That means less people will get it. Okay, amazing. On to the next one. Right, this is one you got to think about here. So some students are investigating the effect of pH on digestion of a boiled egg by using an enzyme pepsin. Pepsin is found in the stomach. It's a protease. It doesn't really matter in this question. You just need to know it's an enzyme. It's a digestive enzyme. So what they've done here is they've put a glass tube and they've put an egg in it. That's the white thing here. They've put boiled egg into this glass tube. And then they've added this enzyme, the pepsin, at pH 7. So this is the liquid here. The gray part here is the pepsin. And what they've done is they set up 6 more of these tubes. So imagine 6 of these tubes next to each other. Each one has a different pH value. So you have one at pH 7, then you have another one pH 6, another one pH 5, 4, and then they do like a whole load of things. And they do the same thing. They put an egg in a glass tube in each of them. And what they do is they leave it for 24 hours and basically see what happens to the egg. Because what's going to happen is the egg is made out of protein, so it's going to get digested and it's going to get smaller. And when it gets smaller, it basically means the pepsin's working. Okay, so the question is, name the product of protein digestion. Good. Turns into amino acids. When you break down a protein, it turns into amino acids, just like we wrote up at the top in our notes. I think we wrote it up here. Yep, we did. There, amino acids. So that's the products of proteins being digested. What type of enzymes digest proteins? Again, it's up there. You can't say peptide, you have to say amino acid. What topic is this? This is it comes under enzymes and digestion. Protease, yep, well done. Not amylase. Amylase breaks down carbohydrates, remember, starch. It's going to finish soon, guys, probably about 10 minutes. Peptide is a bond. It's a bond between amino acids. You don't need to worry about that. Don't worry about that. Okay, next. These are the results. So look, remember I said there was loads of different test tubes. There was pH 1, 2, 3, 4, 5, 6, 7. And what they've done is each glass tube, they've taken out at the end after 24 hours and they've measured how long the egg is. Okay, so you see the lengths here. So at what pH did the pepsin work the best, do you think? People make common mistakes here. People saying 5. 5 is the common mistake people make. It's not 5. It's 2. It's 2. Because remember what it is, guys. This pepsin is breaking down the boiled egg. So if it breaks it down a lot, what's going to happen? The length of it is going to be really small, isn't it? That means it's worked really well. If the length of it is small, it means the pepsin has worked really well. If the egg stays big, it means it hasn't worked at all. Pepsin's meant to break down the pepsin's meant to break down the egg. So the answer would be pH 2. I hope that made sense. So the answer you gave in part BI may not be the exact pH value in which pepsin works. What could the student do to find a more accurate value for pH? Sorry, someone asked to zoom it. Let me zoom this in. Should have done this earlier. So the reason we got 2, again, let me explain again, because that's the shortest length. It's 20. That means it's worked best because the egg has gone from being really long to really short. So that means the pepsin has broken it down a lot. It's broken it down more than all the others. That's why it's working the best there. Exactly. Yes, you need to try more pH values. So that what this question is asking, it's saying it may not be the exact pH because we know it's around 2, but we don't know. It could be 1.5, it could be 2.5, it could be 2.1, 2.2. We don't know, but we know it's somewhere between 1 and 3, right? We know the best pH is somewhere there. We just don't know exactly what pH it is. So what we should do is try more experiments, maybe at 1.1, 1.2, 1.3, then all the way to 2, then 2.1, all the way to 3. So between these ranges, you just want to do more tests. So the answer for this is repeat the experiment with more values between pH 1 and 3. Why between those ranges? Because we know that's where it works best. That's the lowest. That's where it works the best. The egg is the smallest at that. But we never know. At 2.1, it might go down to 19. Or at 2.2, it might go down to like 16. So we need to try all those pHs between to see which one will work the best. This is for any exam board. You can get asked something like this for any exam board. It's just a experiment question. Right. There was no change in the length of the egg white from pH 5 to pH 7. Do you guys see this? It's all stayed at 50. The egg white in each tube was 50 millimeters long at the start. At the end, it's still 50. What does that tell us? Why was there no change for pH 5, 6, and 7? Yes, enzyme denatured. Well done, guys. Loads of right answers. So that's what happens to the enzymes here when they denature. Let me zoom in here. When they denature, so if the temperature or the pH gets too high, then this active site is going to change shape. So it won't be like a T shape anymore. It'll look like that. So now this lock and key mechanism won't work. This won't fit in here. It's permanently changed shape. The active site's changed shape. Substrate is no longer going to fit in it. Why did it denature? Because the pH got too high. Two things affect enzymes: temperature and pH. If temperature gets too high, it'll cause it to denature. If the pH gets too high, it depends when the optimum is. If it gets too high or too low, then it's going to denature as well. Here, what's happened is the pH has gotten too high. Do you see this? The higher you get, the less it's working. And down here, it's completely not working at all. So it must be because the pepsin has denatured. So pepsin denatures. Active site changes shape, and substrate, in this case, in this case it's the protein, doesn't fit in. Alter shape, I guess you can. Yeah, I'm going to let you guys screenshot it in a second, don't worry. Can you see the enzyme denatures, or do you have to say pepsin? Enzyme. You can say enzyme, that's fine. What if you spell something wrong in the exam? Okay, so you can spell things wrong. If you've seen a mark scene before, sometimes it says phonetic. That means if you spelt it how it sounds, if you spelt it wrong, then they usually give you marks for it. But all the key words, try and spell them correctly. It depends on your marker as well. You can use bullet points. Yes, you can. How many marks is this question? 2 marks. 2 marks. Okay. Right. Let's finish this off. So remember, pepsin is made by the stomach. Name the acid in the stomach. Yeah, tomorrow I'll get some questions in the heart, definitely. Hydrochloric acid. Amazing. Amazing. Well done, guys. So, one more day of revision. I'll let you guys screenshot this. But remember, if you want any more, go onto YouTube and just look at the past lives that I've done. If you want more practice on past paper questions and how to answer them, I literally go through them. You can print out each of the worksheets and do them while you watch the videos. So health and disease, homeostasis, nervous system. Those are the relevant ones for bio. So do that, and tomorrow, I'll get some questions on the heart, and I'll try to get some on hormones as well. So take a screenshot of this, guys.